Are you ready?
Yes, Bu Reni.
Yes. Please, for 10 minutes. I hope.
Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. Let me introduce myself. My name is Desi Sasdieta, and I'm from Universitas Negeri Jakarta. I would like to present our paper entitled Screening and Isolation of Indigenous Cellulolytic and Proteolytic Yeast from Fermented Dried cocoa beans. This is our outline of this presentation. First, introduction, and then research methodology, and then result and discussion, and last one is conclusion. First, introduction. Cocoa beans fermentation is a process by which large molecules are broken down into simpler molecules with the assistance of microorganisms. These are one of the microorganisms that mainly responsible in cocoa beans fermentation. They degrade pulp and produce ethanol from glucose under anaerobic and acidic conditions. Yeast has been known to have a lot of potential, such as the potential to manufacture cellulose and protease enzyme. Cellulose is an enzyme that known to degrade cellulose into glucose. Cellulose is often used in biotechnology and industry. Protease or proteolytic enzyme hydrolyze peptide bonds. Protease are also used in the food, bioremediation, and pharmaceutical industry. And main objective of this study is to obtain and test the ability of yeast isolate from fermented Sulawesi 1 and TSH 858 cocoa beans to produce cellulose and protease enzyme. And this research methodology, first yeast isolation, and then yeast colony purification, screening of cellulose, and screening of protease. Result and discussion. First, yeast isolation. The isolation result obtained as many as 231 yeast isolate consisting of 136 yeast isolates from chocolate variety Sulawesi 1 and 95 yeast isolate from chocolate variety TSH 858. Based on the table 1, it can be seen the yeast has grown since the fermentation has not taken place. Fermentation in 0 hours. Next, cellulose screening result. Based on the result of the screening that has been carried out, it was found that 29 isolate from chocolate variety XK and 27 isolate from chocolate variety TK were declared as producing cellulose enzyme. Which isolate were declared positive if there was a clear zone around the isolate. The formation of a clear zone around the yeast colony indicates the activity of the cellulose enzyme that degrades the CMC medium. Talenta et al. Cite that's the size of the clear zone form is an early indication of the presence of the cellulose enzyme produced where the larger the clear zone form, the greater the possibility of the cellulose produced of or having high enzyme activity. The formation of clear zone shows that the cellulose in the medium is hydrolyzed by the cellulose enzyme into a simpler compound, namely cellulose, which is then converted into two glucose molecules. Yes, more at all sites that yeast can decompose cellulose compound and form a clear zone on CMC medium to the process of breaking the glycoside bound in CMC medium by the activity of the cellulose enzyme produced by the yeast. Okay, next, protease screening result. The proteolytic activity assay was carried out using skim milk media. The result of the qualitative test is so that 3 isolate from chocolate variety SK were proteolytic yeast because they were able to produce protease enzyme, which were characterized by the formation of clear zone around the yeast colonized in figure. The presence of clear zone that appear indicate the breakdown of protein molecules contained in skim milk in the media. Proteolytic yeasts are able to produce extracellular protease enzyme, which are protein breaking enzyme that are produced inside the cell and then released out of the cell. Skim milk contain casein, which function as an enzyme substrate. Hydrolysis of casein was used to show hydrolytic activity of the protease enzyme. Based on the result of the test, it was discovered that isolate from Sulawesi 1 in B, D, and E had the ability to produce cellulose and protease. These 3 isolate were isolate after 7 to 8 hours of fermentation. Conclusion, and in conclusion, Sulawesi 1 contained one of 135 yeast isolate known to be capable of producing 29 cellulose enzyme and 3 isolate that have the potential to produce protease enzyme. In TSH858, 27 of 95 isolate were known to be capable of producing cellulose enzyme and none were capable of producing protease enzyme. 3 yeast isolate that were obtained from Sulawesi 1 are capable to produce cellulose and protease. That's all from our presentation. Thank you so much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Ms. Desti Zaziata for the nice presentation. Now we begin to the answer and question and answer session. If you one of your participants can ask, you can raise hand or you can take the question in the chat room. Okay. Maybe the participants still thinking about the question. Maybe I will ask to the Ms. Desti Zazieta. According to the research, the research only take the qualitative parameter to make the conclude the characteristics of cellulose enzyme from the cacao you are collected. Is there any quantitative character that make a conclusion more exactly about the quality of the cellulose enzyme belongs to the cacao? Would you please unmute the speaker?
Kan untuk substansi mah bisa saya tarik. Karena itu kan nggak mungkin di sini enggak. Tadinya kan ada arahannya aja.
Desti? Would you answer my question? Would you please?
Makanya harus
Yes, Ibu.
nanti harus buka komputer
Sorry.
dulu. Ngeling dulu.
Miss Siti Nur Maliati, would you please? Okay, it is already turn off the feed speaker.
Yeah. Sorry, Bu Reni, can I answer in bahasa?
Okay, it's okay.
Ya, kalau berdasarkan riset ini tidak ada data parameter kuantitatif lainnya, Ibu.
Okay, this from this a part of the research, the team researchers conclude that isolasi and identifications only qualitative data from the in vitro testing, yeah.
Ya, Ibu.
Okay. One more question. What to do next from the following from this research after the team screening and isolation indigenous cellulosic? What to do next? Is there any research to use the cellulosic enzyme that you are that your screen, yeah, or your isolation to the cacao bean? The unmute, would you please unmute the speaker? Do not turn off. Mila Desti.
Bisa, Ibu. Jadi melakukan optimasi media untuk memperoleh hasil yang lebih baik mungkin.
Okay. Okay, thank you for the answer. I should give the applause to the presenter. Thank you very much. Desti Sajeta, you can close this presentation.
Okay, thank you for Bu Reni and all. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Desti Sajeta. For the next presenter, this ABS 85 about effect of yeast consortium on chemical properties of fermented dried cocoa beans will be presented with Mrs. Dahlia or Ms. Proborini.
It will presented by Salsabila.
Oh, this presented by Salsabila Fauzi Mahfuz.
Yes.
Okay, thank you, please. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. Before I start, I would like to introduce you myself. My name is Salsabila Fauzi Mahfuz, and I will be the representative of this research team to present this research paper entitled Effect of Yeast Consortium on Chemical Properties. Hello and greetings to everyone. Before I start, I would like to introduce you myself. My name is Salsabila Fauzi Mahfuz, and I will be the representative. Hello and greetings to everyone who've attended.
Excuse me, for Miladia Samsu, would you please help me to arrange this presentation? Or Salsabila, could you please present this by your own? Salsabila is not here.
Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. Before I start, I would like to introduce you myself. My name is Salsabila Fauzi Mahfuz, and I will be the representative of this research team to present this research paper entitled Effect of Yeast Consortium on Chemical Properties of Fermented Dried Cocoa Beans Theobroma Cacao L. And I will discuss the introduction, the research methodology, the research, the results and discussions, and also the conclusions of this research paper. Introductions. Cocoa beans are known to be the main ingredients in the process of making chocolate. There are several cocoa-producing countries in the world, and Indonesia is one of them. However, cocoa beans in Indonesia are generally produced from form dry cocoa beans without a fermentation process. In this case, because cocoa beans fermentation process took a quite long time, and farmers in Indonesia does not want to do that. And fermented and introduction. Cocoa beans are known to be the main ingredients in the process of making chocolate. And there are several cocoa beans producing countries in the world, and Indonesia is one of them. However, in Indonesia, the cocoa beans are generally produced in form of dried cocoa beans without the fermentation process. It is because the process of the fermentation took quite a long time, and the farmer does not like to do that, and they just sell it in form of dried cocoa beans without fermentations. But in several farmer, they do a fermentations process, but carried out in a spontaneous way. However, the spontaneous fermentations of cocoa bean does not always provide a good quality of cocoa beans and can lead to unwanted mold growth and can cause loss of taste, spoilage, and the presence of mycotoxins. And the fermentations of cocoa beans can be assisted by the use of the starter from the microbial cultures, and one of the microbes that can be used is yeast. Yeast is known to be the dominant microorganisms at the initial time in the cocoa bean fermentation process. In the fermentation process, yeast can utilize sugar and pulp, which can produce ethanol, and with the help of enzymes to degrade pectin contained in the pulp. And the protease activity produced by the yeast can also be responsible for producing the cool taste of cocoa beans. And several yeasts are commonly found in spontaneous fermentations, and a lot of them can be used as a starter culture to do the controlled fermentations. And the composition of yeast in fermentation is known to directly determine the development of aromatic flavor of chocolate flavor. The use of starter cultures in cocoa fermentations The use of structural cultures in cocoa fermentations has been carried out in the middle of the 20th century. Since then, different microbial species have been proposed as a candidate starter culture strains, especially yeast microbial species. Based on this background, the study aims to determine the effect of the combinations of the yeast consortium on the quality of the cocoa beans by evaluating values of the temperature, pH, total polyphenol, antioxidant activity, reducing sugars, and the fermentation index. Next one is the methodology. First one is sample collections. We collect these dried cocoa bean samples from Pusat Penelitian Kopi and Kakao Indonesia, Pusat Koka. Then we rejuvenate our yeast isolates. In this research, we use four types of yeast isolates, namely Pichia crudeus ZPE, UNGCC Y156, we namely code A, and the Pichia scutulata, UNGCC Y144, we code it B. The third one is Saccharomyces cerevisiae, UNGCC Y87, and we code it C. And the fourth one is Pichia sesembensis, UNGCC Y157, and we code it D. And after we rejuvenate the yeast isolates, we prepare the yeast starter. And then after we have the starter of yeast, we ferment the cocoa beans and inoculating the yeast starter. And then after the fermentation is conducted, we do some analysis during the fermentations. We do some analysis of pH, the temperature, reducing sugars, total polyphenol, antioxidant activity, and the fermentation index. This is the results and discussions. First one, there are the temperature. Temperatures and pH are the important factors for evaluating the progress of fermentations. Temperature itself is an indicator of the success of the fermentation process, and naturally, the fermentation process was initially characterized with slowly increasing temperature, and in the middle it experienced a drastic increase and became static towards the end of fermentations. And the result of the study on the changes in temperature during the 48-hour fermentations with the 24-hour intervals on the 6 treatment combinations showed that the temperatures increased slowly and the first day and then increase again on the second day of the fermentations. And the increase from the 24-hour to the 48-hour reached the temperature of 29 until 35 degrees Celsius. Second one is the pH. And the pH of the cocoa bean is an important physicochemical properties because it gives indications of the degree of the acidification of the cocoa beans during fermentation and the quality of the fermentations. In this study, the pH of the combinations of the yeast consortium of code ABD and the ABCD had a pH at the constant rate increasing during the fermentation process with the final pH value of the fermentation 6 and 7. This can happen because yeast break down the sugars and make some citric acids that can cause the increase of the pH accompanied by an increase in temperature due to the heat that arises in alcoholic fermentations. And the condition at the stage of optimal fermentation condition for growth of another microbial in anaerobic state. And the next one is reducing sugars. In cocoa beans, the sugar content in the beans is sucrose, glucose, and fructose, and sucrose is the main component, and followed by fructose and glucose, which make up about 6% of the total sugars. Based on the result, the study, the reducing sugar content of the combinations of inoculum in code ABC, ABD, ACD, and BCD, and a negative control increased until the end of fermentations, with reducing sugar level ranging from 5.1 to 6.7 milligrams per grams in the figure number 3. Where the combination treatment of ABD inoculum experienced an increase in the highest reducing sugar content of 4.5 milligrams per grams from 2.3 to 6.7, and this is in line with the research of Afu Aqua, where the reducing sugars content increase in the end of fermentations. And reducing sugar is the precursors of the carbonyl aroma in fermented cocoa beans, which is mainly produced by hydrolysis of sucrose and fructose and glucose. The increase in reducing sugars concentrations with may be due to the hydrolysis of the sucrose into glucose and fructose by the invertase enzyme. And however, reducing sugars can also be formed by enzymatic hydrolysis of anthocyanin to yield arabinose and galactose by the actions of the glycosidase enzymes. And the production of the reducing sugars during fermentations is very important because these sugars will react with peptides and free amino acids in the Maillard reaction during drying and roasting to produce the characteristic of cocoa flavor compound. The next one is the total polyphenols. The cocoa bean itself, the polyphenol of the cocoa bean itself is comprising 12 until 18% of the total bean weight and have long been associated with taste and color of the chocolate. And based on this result, the treatment of ABC, ABD, ACD, and BCD has increased in the first 24-hour fermentations and then decreased in 48-hour fermentations. Meanwhile, the ABCD and negative control has decreased in the first 24 hours and increased in the end of the fermentations. The total polyphenol levels with the highest increase were recorded in the BCD treatment, which was 23.7 milligrams per grams, and the total polyphenol content with the lowest increase was found in the ABD treatment, which was 22.03 milligrams per grams. And the results of this experiment are in agreement with reports of fermented beans where fermentation led to an increase in phenolic contents of seeds such beans, okra seeds, and coffee. In their natural form, phenolic compounds are combined or bonded with sugars, which reduces their availability to organisms. During fermentations, proteolytic enzymes from starter organisms hydrolyze phenolic complexes into simple and freely soluble phenols and more biologically active forms, which are readily absorbed. Next one is the antioxidant activity. Based on the result of this research, the antioxidant activity test, there were two trends in the yield at the end of fermentations, namely an increase and an decrease of the percentage of antioxidant activity. The percentage of the antioxidant activity in cocoa beans with the combinations of ABC, ACD, and BCD increased their activity antioxidant as much as 0.024%, 0.033%, and 0.027% respectively, while the percentage of antioxidant activity within combinations of inoculant ABD, ABCD, and negative control decreased antioxidant activity. Factors that affect the antioxidant activity of fermented cocoa products are the breakdown of polyphenol components, which are influenced by the microflora and temperature and time of fermentations. In this study, the factors that are thought to be the most dominant are the conditions of fermentation time and the condition of the microflora and the quality of the cocoa plant use. This is in line with some research. The longer of the fermentation treatment time, the less the antioxidant activity. And the BPPH radical inhibitor activity decreased with longer fermentations, which means that the decrease in antioxidant activity was due to the decrease in the polyphenols. And polyphenols are the main components of the cocoa beans that play a role in antioxidant activity. And during fermentation, the decrease in polyphenol content was also due to the biochemical modification through polymerizations and complexations with proteins. It also can reduce solubility and the astringency of the cocoa beans. And the last one is the result of the fermentation index value. We're seeing from the ratio of the absorbance ratio of 40. Next one is the fermentation index. With all of the three combination treatment of this research, all has increased the value of the fermentation index and 24 hours of the first fermentation time. And in this result, the combination of BCD and ABD had the largest fermentation index value of 1.16 for the combinations of BCD yeast, 1.11 in ABD yeast. The last one is conclusions. Based on this result of this old study, final temperatures of the yeast treatment was average from 32 until 33.6 degrees Celsius, and the pH value was constantly increasing in the combinations of ABD and BCD with the pH value of 6 and 7, and combinations of ABD treatment also obtained the highest polyphenol content, and it is increased the reducing sugar at the value 6.7 milligrams per grams, and total polyphenols value 23 and 0.18 milligrams per grams. And the lowest antioxidant activity was obtained in the combinations of ABD and ABCD. And based on these results, we conclude that the treatment with ABD yeast combinations has the best fermentation stability. In addition, the fermentation process using dry yeast provides faster fermentation results using 48 hours of fermentation times. I think that's all from our presentations. I would like to say thank you for your attentions. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Waalaikumsalam warahmatullahi wabarakatuh. This is a long presentation. Ms. Salsabila is there in this room? We begin to question and answer, or we begin to this discussion session. Is there Mrs. Salsabila? No, is our the team?
I'm sorry, Salsabila is currently unable to attend this meeting.
Yes.
Maybe I can speaking on behalf of Salsabila.
Okay, maybe our participant will ask the question for the Salsabila and team presentation. maybe I want to ask for the team. According to the data, if I'm not mistaken, about pH, temperature, there is no significant difference between the consortium. Yeah, according to the graphic the team make. And the first question like that. Kemudian the second question about the phenolic compound. There is a fluctuative data in the presentation that in the 24 hours, and then the second 24 hours, the decrease of phenolic compound. Would you please explain for the first question, how the team makes a conclusion that the best temperature or the best pH to make the good fermentation in this research? So what the temperature and pH that you make a decision to make the best fermentation for this cacao? The team maybe could answer. Roborini or Axel, I think this is the one of the team. Desti also, Almira also, the team. Okay, unmute the speaker. Please answer because the time is running.
Can I answer the question?
Yes.
We make a conclusion with the best pH and temperature. We connected with the fermentation index,
Okay.
Fermentation index that the result is nightmare.
Okay, according to the data, ABC, CD, then something like that. There is no different effect temperature. So you make a decision that for what fermentation index to make the conclusion for this research. Is it right?
Yes, ma'am.
Okay, Vector. Of course. How about the fluctuative phenolic compound from this research?
I think we think the phenolic compound is different because when we use the yeast isolate, there's a different activity because
because of ability of the consortium to make the secondary metabolites of the cacao, maybe, yeah. Okay, thank you for the answer. This is the second session. Alhamdulillah, you can make a closing presentation. Maybe your Proborini will represent of your team. Okay, we make a close to this session. Okay, Alhamdulillah. Thank you very much for your presentation. For the third presentation, we'd like to invite Mrs. Nifasini Nela Mikan to present the screening of factor influencing riboflavin production by recombinant ASP, COCP using Plackett-Burman fractional factorial design experiment. Would you please, Mr. Nifasi, time is yours.
Hello and greetings to everyone who've attended today. Myself, Nivashni Nilamegan from University Technology Malaysia, would like to present on my research topic entitled as Screening of Factors Influencing Riboflavin Producing Recombinant Ashbia gossypii using Beckett-Berman Fractional Factorial Design Experiment. Now, the riboflavin is one of the water-soluble vitamin which is physiologically needed by the body in order to undergo cellular metabolic reaction. But it is only possible when we have fulfilled the dietary need of it. So the changes of dietary intake is basically driven by the increment of world population, the cultural differences between a certain ethnicity or a group of person, medical and health concerns of a certain people, as well as nation situation towards providing a sufficient food supplementation across the country. So the dependency towards natural food is very much questionable, and nowadays people are very much preferable towards functional food derivatives such as riboflavin. And this has actually heightened the world market demand of riboflavin, apart from the intensive application of riboflavin in various industries and health growth patterns. So we have approached to use a microbial production for a large-scale cultivation of riboflavin. In that, Ashbia gossypii plays an excellent role of riboflavin producers, as it has a special tendency to overly produce riboflavin, but about 40,000 times greater than normal growth requirement. By using this non-synthetical route and recombinant strain of Ashbia gossypii, we able to enhance the productivity in a long run with minimal range of side effects. The reason we have chosen fermentation, which is the non-synthetical route, instead of chemical synthesis, is because of the drawbacks of chemical synthesis, such as the waste disposable management and the output of waste more than the product. The aim of this study is particularly to optimize the medium composition that efficiently produces riboflavin via statistical approach. A part of this statistical approach is where the statistical screening comes in, which is known as Beckett-Berman Design Experiment. Beckett-Berman Design Experiment is way more different than the classical method because it only studies a set of variables to determine and influence variable which have affected the riboflavin synthesis. Unlike that, OFAD method is more likely to be studied to understand the interaction that exists between the variables to provide an efficient riboflavin production. Now, I would like to emphasize onto the figure at the left-hand side where it has shown that fatty acids has elevated the productivity of riboflavin. Where does these fatty acids come from? Well, by applying different varieties of oil component that composed of triglycerides, these can happen where triglycerides being hydrolyzed by the enzyme synthesized by Ashbia gossypii. The enzyme that's synthesized by Ashbia is known as lipase. It hydrolyzes triglycerides into free fatty acids and elevating the productivity of GTP that simultaneously produces riboflavin. Free fatty acids are being ingested and these are being stored or accumulated as lipid form and further being produced as a riboflavin. So whenever we use a lipid substrate, this glycosylate cycle will be active and it enables us to bypass and produces riboflavin in more efficient way. So other than any oil substrate and other than oil substrate, it depends on the enzyme that it produces and pathways that are available for it to undergo metabolic reaction and produces riboflavin. So that is why oil-based substrate are being the core of this study. This will be the whole procedures of our experiment, starting off with the establishment of cell bacteria analytical part. We are in the stage 2 shake flask studies. basically before the response surface methodology to optimize the medium. In stage 2, we have done spore initiation and isolation to establish our master cell bank. This spore being utilized to cultivate on a culture composition, which also have done a screening procedure as well. This is how it looks on a solid medium. The result over here have shown that the solid media screen that is suitable for the culture to grow will be media 2 with highest radial growth and very good topological characterization, where in terms of the mycelium that it produces is very much intense. The more intense it is, the higher the riboflavin productivity. This particular culture were actually used to prepare our inner clump. Inner clump that inner clump itself has 2 stages where we have to prepare in pre-seed for 3 days. These pre-seeds contain yeast and dextrose, and the mycelium that it produces is very much visible compared to the seed medium. When we view under the microscope, the filaments tend to be entangled, and the seed medium, it is not that visible. The composition of seed medium is constick liquor, yeast extract, and oil substrate. This was cultivated for 24 hours only before being transferred into production medium for further experiment. And then we have done screening, liquid media screening, when we found that media 2 is the best one to be depend on for the cultivation of riboflavin. It has given about 3.77 gram per liter of productivity. However, the challenging part over here is the composition of gelatin and glycine. Since gelatin is an animal source and most of the amino acids that it contains are not that essential, also the amount that we are using over here is about half of the nitrogen source. We thought of replacing with other type of nitrogen sources or maybe remove it from the composition. Glycine, since it is important for riboflavin synthesis, we thought of reducing the amount only. So half of it, half of the amount only we are actually using. And after we have done this improvisation, the productivity has reduced to 2 gram per liter, which you'll be seeing at the end of the slide. This is the whole improvisation which we have done using the OFET method. Basically, the carbon source being replaced with sunflower oil, and there is an extra nitrogen source we have added to it, which is yeast extract, but in the least amount of it. And surprisingly, amino acids that is suitable for the productivity of riboflavin, each is half of the amount from the original composition. Unlike the rest of the element, for mineral source, it is a different case of scenario where without the amount of potassium phosphate, it has given a better productivity of riboflavin. But when it comes to industrial scale of cultivation, we still have to consider the amount of minerals that we wanted to add on the composition. So in here, we have chosen the second best of concentration, which is 0.5 gram per liter. And there is a new element we have added over here, which is biosurfactant, in order to emulsify the oil substrate and further facilitate the movement of lipid to produce riboflavin efficiently. So over here, we have chosen twin 60 in 1%. This is how it looks after the media being optimized using classical method. For the statistical screening, all the elements, all the variables have been screened, and we found that only yeast extract have not passed the reference line. This Pareto chart have sorted the chart in descending order. It shows the cruciality of a particular component. So from here, we can say that sunflower and biosurfactant is much more needed for the riboflavin synthesis. As for the normal plot, it a bit similar to the Pareto chart as well, but only in this case, as the farther the dots are, the crucial, the more important the particular variable would be. So in this case, again, sunflower and biosurfactant, which has given highest percentage of impact towards riboflavin productivity. And in the ANOVA table, we can see clearly that all the p-values for particular variables are lesser, except for the yeast extract, which has given 0.895. Now, this model have wholly shown that that it has significantly impact towards riboflavin synthesis. And the summarized table have shown over here that the productivity of optimized medium, the one that screened with statistical approach, has given more productivity compared to the unoptimized one. As you can see, when we have removed the gelatin and reduced the amount of glycine, it has dropped to 0.2772 gram per liter. As soon we have introduced yeast extract and replaced the carbon source with another alternative and biosurfactant as well, it has increased up to 0.6284 gram per liter. This.
Okay, thank you, Mrs. Nifas, Sini, Nela, for the interesting presentation. Now we begin to the discussion section. Is there any question to the participant? Yes, still is quiet. Okay, maybe Miss Dalia Sukmawati, maybe you will have a question. It's not. Okay, maybe I will ask her. Can I ask you something? Okay, thank you very much. This is very interesting. Yeah, you use Burman fractional design experiment. What's the difference between this design experiment with ANOVA with the much factor? Yeah, according to the data, there is no significant products use yeast extract. Yeah, there are more than 0.85. Yeah, what is the conclusion? Is there we can use that yeast extract from this research?
Okay, thank you for the question. So according to the ANOVA, as you can see, the p-values, anything that is lesser than the p-values, which is 0.05, is considered significant for the riboflavin synthesis. So the one that have more than 0.05 value, it's considered you can use the original concentration value, which means before this, after we have done the off-fat optimization method, we found that yeast extract, the suitable amount of yeast extract is 2 gram per liter. So when we have again optimized using the plaquette Burman, we found that yeast extract only have not passed the p values. So from that we can conclude that anything that more than 0.05, we can stick with the original concentration, which is 2 gram per liter. No need to further optimize and use response surface methodology to further study the different type of concentration for respective element.
Okay, I see. So if we will know that recombinant SPI-CPE, what the gene that you introduced in this SPI-CPE is from yeast?
Actually, this particular genetic material we didn't modify it. We obtained this particular strain from Japan, from Shizuoka University.
Oh, I see. This is you already have the recombinant this yeast, and then you the team make it to increase riboflavin in the substrate, and vitamin. Okay. For one question, is there any color chart that you have to make decision that the color is green or orange to the light orange, like something like that?
That is for response-based methodology, which is after this, after plaquette Berman screening, only we will proceed for response-based methodology, which I haven't done.
Oh, I see.
Yes.
Okay, thank you for the this is very interesting. Yeah, you can increase the riboflavin vitamin using the recombinant SBA-GOSP. Is that true? Okay, thank you. That is a very interesting presentation. You can close the presentation, please. Okay, we can applause to this third presentation, and the fourth presentation we will please Ms. Rida Mahyuni to represent Rafflesia in Mount Leuser National Park, Indonesia, on update distribution and conservation status.
Bu Reni, I'm so sorry, it should be Aryani binti Nasir, Bu.
Oh, Miss Rani Aryani. Oh, according to, okay, please, maybe I have a previous abstract. Okay, please, Miss Aryani.
Hello, yeah. So my name is Aryani binti Nasir, and I am from University Technology Malaysia under Department of Bioscience, Faculty of Science, UTM, and I am under the supervision of Dr. Siti Paulina binti Muhammad Buhari, and the title of my presentation is Fabrication and Characterization of Cellulose Acetate Nanofiber Derived from Rice Husk as Potential Dressing Material. So this study will discuss the expressions of the cellulose from rice husk waste and the characterization. So as for the introduction, so diabetes is one of the contributors to chronic wound, and wound care management is essential in treating diabetic wound. So natural polymers or biopolymers are now explored for the fabrications of dressing, and hence I would like to elucidate the potential use of cellulose derived from rice husk to be used and to be explored as a wound dressing. So rice husk is a waste commonly disposed by open burning, and various research have been done on the biomedical applications of rice husk, including in drug delivery as well as in tissue engineering. So incorporations of active compound will assist in wound healing. So active compound can be used, for example, it's like flavonoid, and it will be added due to its antioxidant, anti-inflammatory, antimicrobial, and anti-diabetic properties. So an ideal wound dressing should be biocompatible, and electrospun nanofibers have been commonly used to produce dressing, as the structure is similar to the extracellular matrix of our skin. So it displays biodegradable, biocompatible, and biomimetic properties, as well as it is tunable. So next, on the rice husk. So rice husk is one of the major agricultural waste, especially in Asia, and it is commonly disposed by open field burning, which will be detrimental to health. So the researchers are now exploring the potential use of cellulose from rice husk due to its growing demand on the exploitations of its cellulose content. So rice husk consists of around 35% of cellulose, and cellulose is a very good candidate for wound dressing. So it will be fabricated to be used as wound dressing because it is based on biocompatibility, biodegradable, and it has cytocompatibility with human skin fibroblast cells as conducted in vitro study. So the significance and the novelty of this study is, okay, so the significance is to understand the benefits of agricultural waste derived fabricated which will elucidate the mat's potential use as an ideal wound dressing. So the nanofiber mat is expected to release the active compound in sustained manner, which is very beneficial, especially for chronic wound. So apart from being cost-effective and environmentally friendly, so the mat is expected to show cellular compatibility and hastening wound healing process. So this study will be used as a stepping stone for further improvements in the future. So this is the problem statement and the objective of my study. Okay, so currently, for those who have chronic diabetic wound, they have altered wound healing in which there will be prolonged inflammatory phase. And currently, there is no specific wound dressing tailored for diabetic wound. And hence, the objective of this study is to synthesize and characterize the cellulose fiber extracted from rice husk to produce a nanofiber mat and to fabricate and characterize the physical-chemical properties of the electrospun nanofiber mats, as well as to look at the cytocompatibility of the fiber with the fibroblast cells. So this is the illustrations of my project in which the cellulose from rice husk will be extracted by classical chemical treatments, and it will be fabricated by electrospinning process as shown in number 3. So this study could potentially improve cell viability, cell migration, angiogenesis, and it will also displace antibacterial and antioxidant properties to stimulate angiogenesis and to accelerate diabetic wound healing. Moving on to the methodology of this study. So it will be divided into 2 stages. In stage 1, so basically this is a series of chemical pre-treatment that was done in chemical lab in UTM. So firstly, rice husk will be washed, it will be dried, and it will be weighed up to around 10 grams. And they will undergo a series of acid alkaline treatment before bleaching. And then the purified cellulose will undergo cellulose acetylation. So cellulose acetate is the derivative of cellulose that is commonly used as wound dressing. And in the second stage, we will undergo electrospinning process, okay, in which there are certain parameters that will be controlled, such as the concentrations of cellulose, the voltage, the flow rate, and the distance from the syringe to the collector. Okay, so this is a short video on the electrospinning process, and the fibers will be collected on the aluminium ball. Okay, so this will be the series of characterization testing that will be done. So as I mentioned, the parameters such as flow rate, distance, percentage of cellulose, and voltage will be monitored. And then characterization process such as fiber diameter, scanning electron microscope, and water contact angle will be discussed later. And as for the results and discussion of the study, so these are the parameters that were optimized. So for parameter 1, we can see that the fiber form on the aluminium foil is not smooth and is not continuous. So we improve the parameter, we optimize, and for parameter 2, we can see that the nanofiber collected on the aluminium foil is smooth and continuous. So we increase the voltage and we reduce the flow rate, and that improves significantly the appearance of the nanofiber collected on the aluminium foil. And after that, we compare the percentage of CA and we observe the appearance of the nanofiber under scanning electron microscope. So for the CA concentrations of 15%, 16%, and 20% under 1,000 time magnification, so these are the observations. So we can see that for 50% and 16%, we are able to produce the nanofiber, but it doesn't look as smooth and as continuous as 20% cellulose acetate. So we further characterize by doing the water contact angle to look at the wettability of the nanofiber. So from the result here, we can conclude that as the concentration of cellulose acetate increase, the water contact angle decreases. So from here, we can see that the nanofiber displays hydrophobic properties. So we want the wound dressing to be slightly hydrophobic so that the drug or the active compound can be released in sustained manner as we are targeting for chronic diabetic wound. So if it is too hydrophilic, so it will be a burst release and it is not tailored for chronic wound. And hence, CA20 favors the wettability, and we can say that CA20 has the highest water penetration rate and favors to be used as scaffold. And as for the fiber distribution, so basically this is the SEM results at 2000 time magnification for CA20. Okay, so the fiber distribution appears within 200 to 400 nanometer. So this fiber distribution is similar to that of the extracellular matrix of our skin. So it will enhance adhesion, proliferation, and infiltrations of human skin fibroblast cells, and it is suitable to be used as dressing. So as for CA15 and CA16, it doesn't show better distributions of fiber compared to CA20. So this table 1, summarizes the physical-chemical characterizations of the electrodes.
Okay, thank you very much, Ms. Aryani. This is very interesting. This is not my background education because I'm studying about agronomy, but I'm very interested with your presentation. As we know that, maybe the participant will ask to Ms. Aryani a question. Maybe it is very difficult research. Okay, this is interesting that usually rice hulls we use as a media for the growth of plant, but this is not a good because there is maybe too many fiber, so they can absorb nutrition in the media. So I'm asking how many rice hulls to produce CA, to produce fiber, to make comparing. Maybe I will know that how many rice hulls we need to have the potential fiber to use as a diabetic. Okay, is that true? Yeah.
Okay, yes. Okay, so first of all, thank you. Okay, so basically from the raw rice husk, so firstly I will weigh around like 10 grams of the raw rice husk, and then from there I will use like series of chemical reactions. So it will undergo like alkaline process, acid process, and bleaching to make sure that we get a very purified, a very clean and purified cellulose microfiber. And then after that, we'll undergo cellulose acetylation. So basically the end product will be around only like 2.5 grams. So after all of the chemical series of chemical process, so we'll have only around 2.5 grams. And then that amount of fiber will be, that amount of rice husk will be added with the solvent for electrospinning. Okay, so we'll add the solvent, acetone and DMAC, and then only we will undergo electrospinning process like what I've shown just now in the short video, and then we produce the nanofibers. So basically we will get around like one piece of nanofiber from that 10 grams of raw rice husk.
Oh, I see. This is, I think this is significant, yeah. From 10 grams, we have 2.5 grams nano fiber.
So yeah, around.
This is my curiosity. How we use this fiber to the diabetic patient?
Okay, so basically that nanofiber it's just very thin fiber.
Thin,
So
yeah.
that fiber will be added with certain drugs. So like in my study, I'll use camphor, which is a type of flavonoid that we can commonly found in plants. So they have like very rich antioxidant, antibacterial properties. So we will fabricate together the drug during electro spinning, and we will directly apply on skin. But since it is biodegradable, so it will just degrade by itself. So we don't have to like remove. Like when we use like Handy Plus or anything, like every few days we need to remove, we need to replace with new wound dressing. But for this one, since it is cellulose, it is biodegradable, so it will degrade by itself, so we don't have to replace. So like for diabetic wound, it will be very painful and it will take a long of time for the wound to recover. So by using this nanofiber, it will ease the pain and it will be convenient for the diabetic patient.
So
that's why we are interested in biopolymer or natural polymer.
Yes, this is very interesting. So we can we put the nanofiber in the lesion of the diabetic without the okay, that's very interesting. Okay, that's maybe we will have a paper. I will read for the whole paper. This is very interesting because we can use the waste product of the rice husk,
and convert it into biopolymer, which is very useful for treating wound.
Okay, alhamdulillah. Thank you very much for
thank you. Okay, assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. For the fifth, Miladia, maybe you can help me. Muhammad Isnur Nur, ya? Oh, Ibu Rida. Okay. Welcome, Ibu Rida Wahyuni. Please.
Ladies and gentlemen, I'm Rida. Today I'm going to talk about Rafflesia in Malaysia National Park, Indonesia, and updated distribution and conservation status. I'm Rida Mahyuni. My colleagues Sadwina Hamamadalimte, Yusana Eritonga, and Yayan Wahyu Chandra Kusuma. You are from Research Center for Biosystematic and Evolution. It's a Center for Ecology and Ethnobiology, National Research and Innovation Agency.
Ibu Rida, is there any problem in the sound because I cannot hear the voice? Or it's just actually only the slideshow? Ibu Rida.
Sorry, Bu Reni. I'm in airport right now, so I have a problem to hearing your sound and many sound in around.
Okay, this very noisy, Ibu Rida. Okay, thank you. Is there any question for the participant?
I apologize because my video very slow because the previously I have a technical problem to upload the all the of video.
Okay, doesn't matter.
If any question, we can discuss about it.
Okay, thank you, Ibu Rida. This is a very fast, very fast presentation. I not reading quite better. According to Rafflesia, is there how many Rafflesia found in Mount Lester National Park? For the first, is there any different maybe variety or Rafflesia wild species? Yeah, I think wild species.
Yes.
Is there any any different between variety? Is there any different variety that the team found in the Mount Lester National Park, or there's same variety?
Okay, there are 3 species in Mount National Park, Rafflesia, and all the species are endemic only Mount Lester National Park.
Okay.
And around the 9 species in Sumatra, and these 3 species in Mount Lester National Park, only this area, and we cannot find another location.
Okay, I think this is the problem for conservation, the Rafflesia, because only endemic. Maybe we can in situ conservation, maybe for the conservation Rafflesia. Is there a lot of Rafflesia, Ibu Rida? How many Rafflesia found in the?
In Indonesia, we have more than 13 species.
Oh, I see.
In Indonesia, in the world, we record more than 30 species. Philippines, Malaysia, Thailand, 1 species, and in Indonesia.
Okay, that's
and in Indonesia, we have distribution Sumatra, Java, and Borneo.
So
I think for the Institute Conservation, start the last year we start to study of ecology, and this year we start to study genetic population.
Yeah.
And until now still on progress to analyze data.
Okay.
And we have some project in another island in Java and Kalimantan of this years. Maybe we invite to collaborate to the stakeholder and participant here to we can to do what and we can collaboration research to conservation of this Rafflesia in Indonesia.
Yes, this is very interesting. Okay, in biology study program, I'm maybe Pak Agung will I will ask Pak Agung to contact Ibu Rida. This is one more question. Is there any chance that Rafflesia not conserved in situ? Maybe for we use tissue culture, is there any chance to make Rafflesia more produce more plant? Is there any reason?
Part of our project, we try to increase in situ conservation and ex situ conservation. We start in this year in population Rafflesia joliniana in Banyuwangi, East Java. We try to propagate the seed in the population, and we try to bring the house in Purwodadi.
Botanical Garden Purwodadi, ex situ conservation, yeah.
As we know that Kebun Raya Bogor successful to ex situ conservation of the Rafflesia fatma, and we try to in another location to do that.
Okay. Thank you very much, Ibu Rida, for the interesting research. Alhamdulillah, for the next presentation.
Dari situ, Bu Reni, one other question in chat.
Okay. Oh, I'm sorry. Professor, may I ask what's the main difference in morphology for each Rafflesia species in Mount Laser? I apologize, Ms. Rida. There is one more question. Miladia asking for the main difference morphology for each Rafflesia.
Yeah, thank you, Bu Reni. Yes, the 3 species of Rafflesia in Mount Laser National Park and 1 species are not cited until now. Just 2 species we kind of found this location. The first Rafflesia longensis has a unique character and difference with another Rafflesia in the world. Rafflesia longensis have the large opening diaphragma and smooth on the diaphragma. Jadi and another species Rafflesia microflora, the small aperture of diaphragma, and this very unique character, and when we distinguish of the other species. And Rafflesia longensis or absent the windows, and as we know that mostly of the Rafflesia have the character like white on the inner diaphragma, and but Rafflesia longensis without the windows.
Okay. Yeah, thank you. Maybe no, I'm studying about the banana. One question that INIBEP pronounced that we have to describe according to INIBEP in Rafflesia. Is there any book description to categorize, or maybe breed already published about the, if we want to go to somewhere and we found the Rafflesia, so we can identify the variety of the Rafflesia. Is there any description, morphology?
Yes, we have some journal to check of the description. In of Rafflesia longensis, we published in 2010 as a new species, and Rafflesia microflora published by Mayer, I think 1984 something like that.
Okay. So we can advise our students if they want to describe Rafflesia maybe in the field. Thank you very much, Ibu Rida, for the next.
Thank you, Bu Reni.
Yeah, thank you very much for the next presentation. Isn't it? Miladia, is that?
Hello everyone, I am here representing my team to share our work on the next selection of hawkbill turtle obtained in Thousand Island of Jakarta, Indonesia. The study will present about the assessing the consistency of hawkbill nest selection between local nesting habitat in the Thousand Island of Jakarta and implication for conservation practice. Okay, as we already know that all sea turtles are in protection and continue to experience a declining population worldwide due to so many factors, including species loss and destruction. To recover this turtle, the population of this turtle, conservation authorities need to do something to help this turtle out. And we assume that protecting and enhancing nesting beaches habitat perhaps are the best action that we can do for sea turtle, since it is easier to manage and does not cost a lot of money. However, the cost earned to conduct this study comprehensively and urgently required action to be applied have driven conservation scientists to adopt a precautionary approach based on the data available only from a subset population. We know there is a danger inherent to this approach. Why? Because precautionary management sometimes fails to address the local needs of certain target population. Thus, conservation biologists need to consider implementing evidence-based management for turtle, which have no parental care behavior. And if you look at the Thousand Island of Jakarta, which consists of hundreds of low-lying islands, in which some of islands are the nesting habitat of half-bill turtles. However, a complete study on nest selection on that island, on the Thousand Island, has been carried out only in small subset of population. So we can generalize that data to all island in Thousand Island of Jakarta. Just only we already have certainty that the nesting pattern observed in this island is similar to other. So for this reason, we investigated nest site selection by how-built turtle in Thousand Island to provide more comprehensive understanding of regional variation in this species. We carry out a study on two islands in Thousand Island of Jakarta, which are commonly used by how-built turtle for nesting, namely Kayu Angin Bira and Petualang Timur. Nest sampling occurred from August to September 2019. We patrol daily by foot from 16 p.m. until 6 in the morning, searching for female half-bill. For each encounter female that successfully laid eggs, we mark the egg chamber using tape to help us locate the nest in the next morning. We measure bits width, distance to the highest water line, leaf litter thickness, and the canopy cover. We also employ 100 square meter using the nest as the center of the plot to record the number of trees around the nest. We also use smaller plot, 25 square meter and 1 square meter respectively, to record the number of shrub and ground cover around the nest. To assess whether nest selection criteria used by turtle reflect the differences in availability of suitable habitat in both islands, we also measure similar data on pakan site by randomly selecting site located 20 meter from nest site. We compare nest site attribute between two islands. We also projected nest site attribute on biophysical condition for each island to figure out whether nest selection variability, if any, is reflected by the availability of a suitable nesting habitat. We generate 95% confidence interval based on 10,000 bootstrap estimate around median values for all nest attributes. A generalized linear regression model with binomially distributed error term in R 4.1 and 0.2 was used to determine which parameter best predict nesting occurrence. Here are our blending on 9 nest site variables observed and present in comparison according to the islands and nested with the availability of suitable habitat for each island. Among 6 variables observed, most of them are under stabilizing selection, in which turtle in both island, in Kayu Angin Bira and Putuan Timur, tend to choose site at or near population mean, or turtle in both island show similar preference for six variable observed. The plasticity demonstrated in this study is associated with the differences and environmental variation between the two islands. Different from the other six variable, two variable, canopy cover and leaf litter thickness, are assumed to be under directional selection. This variable show different preference of turtle on both island, in which in Putuan Timur, turtle tend to select nest with dense canopy cover over Kayu Angin Bira. And based on 5 predictive model, which have delta IC value more than 7, which indicating model performance, the excellent model performance, based on 5 best predictive model, which is the strongly supported predictor variable that appear in 3 of 5 model evaluated. And the canopy cover and the leaf litter thickness appear in 2 of 5 model, whereas island or set just only appear in 1 model of 5 model evaluated. Other variable were weakly supported predictor variable, which is are not shown in 5 best predictive models. Even though turtle in both islands were shown to have different preference on which parameter, turtle in both islands exhibit consistent behavior in selecting which close to the ocean. The variation in which parameter selected by turtle between two islands is assumed to be associated with slope and proximity to forest borders. Turtle likely choose sites that are invulnerable to inundation by using slope and forest border as indicator. As the turtle perceive the presence of the forest border or steeper slope, they will stop going further and immediately build egg chambers. Thus, the farther the distance between the low tidal line and the forest border, the wider beach profile selected by turtle for nest. And different selection in our study indicate that preference of sea turtle on canopy cover are under directional selection. This finding support the assumption of genetic variation in turtle that shape the preference of nest selection on particular attributes, which is in the study are shown in canopy cover. In addition to genetic variability, different preference could be a result of beach width variation between islands. A wider beach width could have consequence on the higher loss of energy used by turtle to reach appropriate sites that are secure from inundation. If the presence of vegetation is best indicator for turtle to evaluate sites that are sound from inundation and desiccation, then turtle at a wider beach profile would attempt to build nests as close as the forest area. The nests in forest area are quite dilematic for turtle, in which they require more energy to reach that area, including struggling with the many obstacles provided by the forest. Turtle in wider beach profile usually choose nests along the forest border as compensation to avoid more energy loss and by more prioritizing with the proximity to the ocean. Our model show the inclusion of site or island, even though very weak, as well as the supported by the variable of nest selection of turtle in Thousand Island. This finding underscored the need to consider difference in local needs required by turtle with similar life history. For conclusion, we documented behavior plasticity of turtle in Thousand Island, which can be inferred most parameter under stabilizing selection. And this parameter could likely be set using the data compiled from different area. But some parameters which are under direction selection must be considered in conservation strategies and must not be used, must not be set using the data from other islands. So for this parameter, we have to implement evidence-based management to orient conservation strategy.
Thank you very much, Pak Istit, for the nice presentation. For the participant, if you like a question, you can make in the chat or raise hand. For me, this is very difficult research because I'm not research in animal. Maybe just a little question for Pak Isnin. According to the research objective, the team tried to assess consistency nest of the turtle in Thousand Island, Pulau Bira, and the second I forgot. Is there any consistency of the nest of the hawkbill?
Yeah, there is one parameter that consistent choose by turtle to select nest to build nest, which is sand grain size. The other parameter show difference in preference. There is no consistency, but the fashion for I forget. I think six parameter. So the similar trend. I mean for this variable, the turtle choose to select nest in variable which have a values near the population mean. So the difference in this variable is some kind of result from different in environmental condition on which island and Kayuangan Bira and Petron Timur.
Okay, so this is my question. The turtle will come to the nest for the production or something like that. Is it true? If we already categorize the nest of the turtle, so what's the prediction? What kind of the nest that turtles come back atau going forward to the nest? Is there?
All our 2D turtle in Thousand Island choose site with what is it? Wider bit profile, and also have set canopy cover.
Oh, they choose the canopy cover to the breed or something like that. Yeah.
Yes.
Okay. Thank you. Thank you for this other question. I want to ask out of my curiosity, would there be any chance the hawkbill turtle lay in eggs on other island outside the Bira?
Yes. Actually, there are many islands that inhabit by turtle to nest. But the most used by turtle to nest is kayu angin bira, kayu angin bira, Kepulauan Timur, and there is one other island, namely Pulau Sabira. But Pulau Sabira is more interesting island because Pulau Sabira also inhabited by human. So that in Sabira there is human settlement on there, and I think it's very interesting to study the influence of human activity on the nest selection of turtle in Pulau Sabira.
Is it thing? Okay, that's my idea. Answers. Understand what the answer. Okay, thank you very much, Mr. Isnilur, for the presentation. Thank you. For the next presenter, we invite Ibu Tetri Widiani. Is it right, Niladia Samso?
Yes, Bu Reni.
Okay. Ibu Tetri, please.
Yes. Hello, everyone. Let me introduce myself. My name is Tetri Widiani. I'm from Department of Biology, Faculty of Mathematics and Natural Science, Universitas 11 Maret, Surakarta, Central Java, Indonesia. Today, I will present my study, our study. Wait a moment, I will share screen.
Please.
Our study entitled Antimicrobial Potency of Catfish Clarias gariepinus mucus. For the background, catfish or Clarias gariepinus is a culture freshwater fish which is one of the 3 leading aquaculture commodities in Indonesia after tilapia and epinephelus. Catfish is generally consumed as a protein source. The price is relatively affordable when compared to other protein sources. This advantage leads to an increase in the catfish production. Based on the data from Indonesian Ministry of Maritime Affairs and Fishery in 2015, catfish production reached 1.8 million tons, or increase more than 100% from the previous years. In general, we consume catfish in the flesh part. Catfish has mucus, which is a byproduct. Catfish mucus is not widely known for its benefits and has no economic values. Mucus is the first body defense for the fish. Mucus is a physical barrier between fish body and its aquatic environment. Study on various fish mucus showed that mucus contain antimicrobial compounds. They are potential to be developed and applied in human health and aquaculture. However, study on Indonesia catfish mucus or Clarias gariepinus has not been carried out comprehensively. So my objective, our study objective, will determine the protein content of catfish mucus, which has antimicrobial potential using method STS page, and objective number 2 was determine antibacterial and antifungal activities in vitro. Our method will contain 5 steps. For the first step, we isolate mucus from the catfish body. Catfish sample was obtained from the freshwater cultivation unit, or in bahasa unit kerja budidaya air tawar Wonocatur, Cangkringan, Sleman in Yogyakarta. Mucus was isolated from 56 catfishes, range age 1 to 2 months old, and average length 20 centimeters. Catfish were placed in dry condition an hour before mucus isolation. The isolation using spatula, mucus was scraped off the dorsal catfish body surface. And then mucus collection was centrifuged for 10 minutes at 300 and 5,000 RPM to separate the dirt and liquid. The supernatant or pure mucus was stored in refrigerator at 4 degrees Celsius before use. And the second step is mucus extraction. Mucus extraction using glacial acetic acid solvent. In the percentage 3 to 5%. And the third method is electrophoresis. We use sodium dedosyl sulfate polyacrylamide gel electrophoresis, or SDSPH, to estimate the protein molecular weight for pure mucus and mucus extract. In the fourth step, we carry out the antimicrobial activity testing. We use 2 species bacteria that they are methicillin-resistant Staphylococcus aureus, or MRSI, and Pseudomonas aeruginosa ATCC 27853. And for antifungi testing, we use 1 species. It is Aspergillus flavus. In the antimicrobial testing, we determine the inhibition zone around the dish or around the well in the media. And the last method, we apply data analysis statistically and descriptively. And then from the result 1, we characterize the protein from the electrophoresis SDSPH. SDSPH analysis resulted 4 bands, which indicated that catfish mucus contain protein compound. Compare the marker, there are 13 bands, range molecular weight range 10 to 245 kilodalton, as on the figure. 4 bands from catfish mucus have molecular weight 117. In the second protein have molecular weight 58. The third protein have molecular weight 25, and the fourth molecular weight is 15. Among the four band form, a band with a molecular weight of 15.47 kilodalton showed thicker. Yeah, we can see the band is thicker than other bands. It is presumable similar to lysozyme with a molecular weight of 50 kilodalton. Lysozyme is known to have a role as antibacterial because it can destroy bacterial cell walls. The presence of lysozyme in the catfish mucus is in accordance to a previous study that mucus have protein antimicrobial. For the
Ibu Tetri, I apologize because the time is more than 10 minutes. Maybe we will take the discussion session. For the participant, is there any question? To Ibu Tetri. Maybe I will ask, this is, I'm not studying about the microbiology. What I want to know that according to the isolation protein content in mucus catfish, yeah, so this is for the application. If we put the head of the catfish, is there make a conclusion that we can take the proteins that already identified in the mucus, or maybe in another part of the catfish, there is protein content in the part of the catfish. I mean that this is already identified using SDH that protein content already found in the mucus. My question is there any possibility that in another part we found the protein in the same in the mucus?
Yeah, I will answer your question. Yes, SDSH only know the molecular weight, but not identify what is the protein molecules. So we compare with the other study from previous study. From various study, we know that in the mucus contain the antimicrobial peptide or antimicrobial protein. So we know that from the other studies. That's
okay. So if we already know that we can produce or increase, we can produce or increase the protein in the mucus for the next, because the aim of this study is for detect antimicrobial.
Yeah, catfish mucus production is very tergantung.
Depend on?
Yeah, depend on the environmental of the catfish. If the stress condition or infection, it can produce the mucus with different composition.
Okay, maybe the next research, maybe at the next proposal. Okay, thank you. Thank you very much, Ibu Tetri, for the presentation. Okay, maybe there is no question for the participant. We should close the presentation of Ibu Tetri. For the next presentation, please welcome Ibu Tetri Widian. No. Siti Nur Ameliati Triyono. Welcome, Ibu Teti. See you again. Thank you for the in before you're coming to the assessor. Please, Ibu Siti Nurmaliati.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
My name is Siti Nur Amaliati Triyono from Applied Zoology Research Center, National Research and Innovation Agency, GRIN. The research about Trichochlosis haematodus were carried out by me, Rini Rahmatika, Sinta Maharani, and Rahman Effendi. I would like to start to give my presentation with the title Breeding of Coconut Lorikeet Trichochlosis haematodus Linnaeus 1771 in captivity and their optimizing welfare. Coconut lorikeet, Trichochlosis haematodus, is a species of parrot in the family Piscidaculidae. Coconut lorikeet was endangered due to habitat destruction and heavily traded. Their high economic value very often support the illegal trade. The overharvesting indicates significant effects to the decreasing population in their natural habitats. Captive breeding is another solution in order to substitute the wild-caught parrots as pets. Since December 2018, this bird has been included as protected bird in Indonesia. IUCN Red List status is concerned and also included in the list of Convention on International Trade in Endangered Species of Wild Fauna and Flora. The purpose of this study is to determine the crowd size, hatchling success, and fledgling success in the Trichochlosis haematodus, to find out the reproductive health of the species as well as the level of animal welfare. The method, bird care and housing. 6 wild coconut lorikeets were housed in apiary with the measurement 3.90 meter length, time 3.5 width, and time 2.9 meter height, equipped with nests, natural perches, bathing places, drinking places, and feed areas. The study was conducted for a duration of 12 months, from August 2021 to July 2022. Breeding rate, number of eggs, clutch size, number of hatchlings, and fledglings were recorded for estimation of breeding rate. Hatchling success is equal number of eggs hatched divided by number of eggs laid time 100. Fledgling success number is equal number of fledglings divided by number of hatchlings time 100. Method of animal welfare. The method used in analyzing welfare was by assigning a score to each aspect of animal welfare. The score for each aspect was adapted from the regulation of the Director General of PHKA number P6, 2011, which has been determined based on the 5 principles of animal welfare. Assessment was done by giving weight to each aspect of animal welfare. Weighted score is equal weight time score. Final score is equal amount weighted score divided by 5. Result and discussion. Breeding rate of coconut lorikeet in captivity. We got 11 eggs from August until May 2022 from 2 pair bonds and from 2 nest box A and nest box B. Time of breeding potentially breed all year round. Crush size is 1 and 2 white rounded over eggs. Incubation period 23 to 25 days. Hatching success is 81.82% and fledging success is 100%. Chicks fledge out at 60 to 63 days old. Chicks growth and development. Chicks growth and development is from 1 day until 63 days old at fledging. The chick of Trichoceros haematodus is included in the artificial type, which is born with closed eyes. The body is not covered by natal feathers and in a weak condition. So it is very dependent on parental care until the chick can live independently. Chicks will hatch covered in wispy grayish white down. At 10 days old, gray secondary coat starts to appear. Chick will be almost fully feathered at 49 days old. And the chicks will have fledging age at 63 days old. The assessment of the degree of welfare of coconut lorikeets in captivity. There are 5 welfare aspects. The first is freedom from hunger, thirst, and malnutrition, freedom from discomfort, freedom from disease and injury, freedom to carry out most normal forms of behavior, freedom from fear, distress, and other negative psychological states. And the final score is 79.8. The first welfare aspect is freedom from hunger, thirst, and malnutrition. The result of the assessment score for the freedom from hunger, thirst, and malnutrition is 4.5. This score is included in the good category. Some points that are considered were the quantity and quality of diet, drinking, variety of feed types, and feed distribution. The lorikeets consume 79.20% of their diet in the form of siwalan sugar solution, while vegetables and fruits were 20.80%. The second of animal welfare aspect is freedom from discomfort. The result of the assessment score for the freedom from discomfort is 4.0. This score is included in the good category. This result indicates that birds in captivity at Prin Cibinong can live quite comfortably and are protected from physical disturbance. This is in accordance with the existence of a roof in aviary as a shade and protection for coconut loricates from bad weather. 6 wild coconut loricates were housed in aviary equipped with the natural perches, bathing places, drinking places, and feed areas. The third aspect of animal welfare is freedom from disease and injury. The result of the assessment score for the freedom from disease and injury is 3.5. This score is included in the good category. Some of the good points for this aspect include birds in good health. The researchers doing routine checks on the health condition of loricates every day. Monitoring is carried out routinely by researchers every day. Monitoring the rest of the feed, the condition of the feces, and the behavior of the birds to ensure the birds are in healthy condition. Prevention is better than birds getting sick. Regular washing and scrubbing of walls, floors, wire mesh bridges, food and water containers, etc. And also regular sweeping, ranking of scraping, particularly under perches, feeding station.
Thank you, Ibu Siti Nurmaliati. The time, I apologize because the time is up. Okay, maybe is there any question for the participant?
Maybe I want to ask a question.
Yes, please, Pak Isnin.
I think it's good. Okay, thank you for your information. I just want to know, did you observe the fitness of the offspring? Because if we if we have offspring hatch in the apiary, sometimes the offspring will not have a good fitness as when they are hatched in the wild. So I'm just curious, did you observe the fitness such as their capabilities of flying, or maybe about their instinct? In conservation strategies, I think the fitness of offering, especially is about the thing or the organism that we will release into the wild is very important to consider the fitness of the offspring. Maybe that's my question. Thank you, Bureni.
Okay, thank you for the question. Please, Ibu Siti Nurmaliati, to answer the question.
It's not clear. The question is maybe the MC can.
Maybe okay. Maybe Pak Isnin, could you please reply the question about the fitness?
Yeah. The chicks is healthy. They can flying.
They can flying.
Yes, instead of in captivity, but they are very healthy. Because how many times? 3 times, 2 times for each pair bones have laying eggs, and the chicks is healthy until now. And the second pair bones is now already laying eggs again. Yeah. And the parents always take care of the chicks. Very good. Yeah, maybe because in the captivity we give them a good diet for them, so the chicks is very healthy.
Yeah, so about the business is quite good. Yeah, good. I mean the offspring is is in good condition. Okay, thank you, Bu Siti.
Thank you. Is there one more question, Ibu Siti? In the chat, asked about the score for freedom in expressing normal behavior. It's high, but is there any difference between wild and captive loricates?
Okay, thank you. Yeah, because the presentation is cut off. A score for normal behavior is 4.0. That's quite good category because in our captivity, we give them a freedom. The space is quite wide for 6 birds, and we give them with the natural purchase, and we put them with flowers for them able to eat, and they can taking a bath. They love taking a bath, and they can flying, they can running, they can everything they can do for their normal behavior like preening and also walking on the pads. And but the problem different with the in the wild, of course, the food is not so variation with in the wild. In captivity, the flowers only maybe 3 or 4 types of flowers, and the flowers is not so many like in the wild like that. So the time for the looking for food may be very limiting access, not like in the wild in captivity. So why the score is only for 4.0, but still in good category because they can do what their normal behavior. Thank you.
Thank you very much, Ibu Siti. Okay, maybe time is fast up for the question for Ibu Siti. Thank you very much.
Thank you.
Assalamualaikum warahmatullahi wabarakatuh. For the next presentation, we will invite Ibu Nova Ma'arif. Nova. Miladi, is this true? ABS 881.
Yes, Bu Reni.
Oh, I'm sorry.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good afternoon, everyone. My name is Naufal Ma'arif from Biology Education, Faculty of Mathematics and Natural Science, Universitas Negeri Jakarta. Thank you very much for SMIC 2020 for giving me this opportunity to present my research and my college's research. It's about anti-cancer potential of white turmeric extract in MCF7 and HEP2 cancer cells. Okay, we go to the next slide. We know that cancer is one of the 6 causes of death worldwide, with fatalities reaching 9.9 million souls in the world history. In women, especially in women, the most type or the most dangerous type cancer is the breast cancer. So also in 2019, breast cancer is the most cases in the world, until 2.3 million souls cases for the patient. We know that some therapy in cancer treatment, like a chemotherapy or maybe giving some drug, have a side effect. For example, these differs as reduce red blood cells and maybe make the new cancer cell or the onset of the new cancer cells. So the researchers in the world have to discover anti-cancer drugs which has a less effect for the patient. And one of the potential anti-cancer from plant is white turmeric or Curcuma Deduaria. Why? Because white turmeric has the bioactive compound. We know from several research which has an antioxidant, antibacterial, antifungal, anti-inflammatory, and also anti-cancer, but not only from but it's for one cancer. I have checked in the article. It's about for the not for breast cancer, but for skin cancer. And one of the benefits of white turmeric is has a mechanism of active curcumin that is an anti-cancer, can make an apoptosis for and inhibits the proliferation and invasion of the tumor. So the study aim is to test the effect of cytotoxicity of the extract of white turmeric or Curcuma Deduaria in a culture cells, HEPG2 and MCF7, which is a model of liver cancer and breast cancer. The cause that the research choose for this type is because of the highest rate of the highest cause of death from both cancer cells. So the result of the cytotoxicity test of white turmeric on a cancer cell could be and maybe an alternative, the problem of maybe an alternative for the problem of cancer in the world. So for the methodology, this research used an experimental study with a completely randomized design group with a concentration variation. There was 10, 15, 20, 25, and 50 ppm with a negative control cell line without treatment, curcuma Deduaria extract. Measurement parameters include percent inhibition, inhibitory concentration, or IC50, and the observation of cell morphology. First of all, we make an extract from Curcuma Deduaria. It's done with immersion and then for 3 days and compression 3, then the liquid extract is evaporated using rotatory evaporator, and then we get an extract as a colloid extract. The extraction is carried out in the Molecular and Biochemistry Laboratory, Faculty of Mathematics and Natural Science, Universitas Negeri Jakarta. And then for the MTT, say the MCF7 and HEPG2 cells were plated in 96 well plate and then incubated for 24 hours. Then the cells was added by turmeric extract. Then the cell incubated for 37 degrees Celsius for 72 hours or for 3 days, and then added MTT after incubated for 72 hours with a concentration of 10 microliter per well. And after that, the cells were incubated for 4 hours at a temperature of 37 degrees Celsius. For the data analysis, the white turmeric tested using MTT SI get an inhibitory concentration value and then analyze using 1Y ANOVA and then the Duncan test. Duncan test is the test to know the effect of the several concentration treatment. Observation on the morphology is also done for the MCF7 cell line, and we measure descriptively to the dead cells, membrane blebbing, and then apoptotic bodies. And this is the result of morphological changes of cell MGF7, HEPG2 with the concentration. The concentration of extract of the white turmeric are presented in this figure. Here we can know that 1, 2, 3, 4, 5 is representative of the every concentration. 1 is for 10 ppm, 2 is for 15 ppm, 3 is for 20, for 25, 50 ppm. And for the 6 behind my head, I'm sorry, it's about negative control for the there is no treatment in the 6 in the 6 picture in the figure 6. Yeah, we can see that we can see there is that cells. We can see the membrane blebbing and apoptotic bodies, which is the code DC, MB, and AB. And for LC is about live cells. And then it's for the MCF7. in the concentration of the extract of white turmeric. 1, 2, 3, 4, 5, and 6 is the same as a HepG2 cells, which is represented of the concentration of every treatment. Yeah, we can also see the dead cells, live cells, membrane blebbing, and apoptotic bodies. We know that in every figures there is membrane blebbing, there is apoptotic bodies, and there's a cell death, which can we interpret it. The white turmeric has an effect on the inhibition and then the inhibition of the proliferation of cancer cells. Okay, we go to the next slide. Yeah, we can know from this diagram from percent inhibition. According to the result, we know that for MTF7 in the concentration of 50 ppm is 61.86% compared to the HepG2. Yeah, it's only about 18.83%. So it's highly different. Yeah, highly different. And also for the result of IC50 or inhibitory concentration is about 68.58 ppm on the cell line MTF7 and amounts to 40 ppm on the cell line HepG2 cell. So if we know this finding can be inferred by research conducted by Shamsir et al. 2017. If the value of IC50 is an extract is less than 300 ppm, the extract could be an agent chemoprotective against cancer cells. Okay, we go to the next slide. For the ANOVA test, we know that the white turmeric has a significant effect on both of the cancer cells. For example, for the MCF7, it's a significant value of 0 with the alpha is less than 0.05, and then for HepG2 cells, it's also significant values of the 0.002 with the alpha is 0.05. So based on the table, based on the diagram, we can know that we can interpret the white turmeric has an effect of concentration against cells inhibition. And then we also know from the Duncan test to determine the concentration for MTF7 has a different. For example, for the 10 ppm concentration is the for table 2, for the 15 and 20 is table 3, 25 and 50 is for table 4 and 5. So the Duncan test for the HepG2, yeah, I'm sorry. Yeah, we know that there is no different effect, so we can interpret the HepG2 that treated by white turmeric extract has a same effect from the Duncan between 20 and 15 ppm. However, it's a different impact in MTF7. Okay, for the conclusion, based on the MTT test, it was found that the best inhibition results were found as a concentration.
Okay, thank you for the presentation. For the next session, we all apologize that we cut the presentation because this is more than 10 minutes. Thank you for the presentation. And is there any question from the participant? What pathway induce cell apoptotic? Sorry. Ms. Meladia Samsu will ask you about the pathway induce cell apoptotic or inhibited cancer cell proliferation. pathway to induce cell apoptotic or inhibit cancer cell proliferation.
Okay, thank you very much, Bu Reni. I'm sorry I cannot explain it because of the time. Yeah. On, okay, I will answer it. For the pathways, yeah, we know that the best compound from wet turmeric is curcumin. Yeah, curcumin will activating first of all caspase. Caspase is a protein that responsible for the cell proliferation. And also caspase, especially caspase 3, have some pathway that responsible for cancer cell apoptosis. And also it will induce both of the MCF7 and the HepG2 cancer cells. And also another protein that responsible for the apoptotic is from MCF7, especially for MCF7 is BRCA1. BRCA1 is can generate breast cancer, and also caspase 3 cyclone dependent kinase inhibitor 1A or CDKNA1, and also tumor protein or P53. And for the HepG2 pathways, it will induce through the phosphatidylinosytol 3 kinase or PI3K that can inhibit the proliferation of HepG2 cancer cells. Maybe it will answer the question from Miladia.
Is it clear, Miladia? Thank you. Okay, I will ask you, this is my curiosity. How can you manage the viability of the cell cancer in the medium? This is, you have the cell line of the cancer. So that's my question. How you, the team, yeah, make to pretend that the viability of the MCF7 and HEPG2, how make it viable in the medium culture?
From what I know, we can make this viable because of we incubate it. I forgot, in the -20 degrees or degrees Celsius, and also, but after we can make the cell is still alive. We also after we make after we incubate at the -20 degrees Celsius, we I'm sorry.
Sure that this is still viable. Yeah.
Yes, it is still valuable because it's it will storage at the 37 degrees Celsius in the laboratory.
Okay, thank you. Maybe I will just formulate that according to the statistic analysis, maybe the team should calculate one more time, because I see that there is a standard error higher than the mean of the data, and there is a zero data that should be transformed. Yeah, so from the data analysis, you should discuss to the team. I think this is not correct if we comparing the zero data to the other data according to the statistic analysis. Okay.
Thank you very much, Bu Reni, for the advice.
Okay, thank you. Thank you very much. I give applause to Noval for the next presentation. Miladia Samsu, potential analysis of water, Hyacinth, Echoronia recipient as nanocellular mass filter material. Please. Miladia Samsu.
Good afternoon to the Science and Mathematics International Conference 2022 participants and panelists. Allow me to introduce myself. I am Miladia Shamsu from Biology Department of Jakarta State University. In this opportunity, I am going to present my paper with the title Potential Analysis of Water Hyacinth Eichhornia crassipes as Nanocellulose Mass Filter Material. The world is currently experiencing a COVID-19 pandemic that threatens our health. This pandemic is caused by SARS-CoV-2, which attacks the respiratory system. COVID-19 spreads through droplets, physical contact, and surfaces contaminated by droplets from an infected person. The infection can be prevented by using masks, and because of this, the government orders mask mandate to its people. However, this mandate also caused the side effects, which is increased mask waste, because most people still use disposable surgical masks. To combat mask waste, we can opt for cloth mask, which is reusable. But according to study by Captivity et al. At 2022, cloth masks showed relatively poor performance compared to medical masks in clinical trials. So what is the solution? The filtration efficiency of cloth masks can be elevated by placing additional fabric that works as filter. Usually people use tissue, but there isn't any research to prove tissue's filtering ability. One of the solutions is nanocellulose filter. This is because nanocellulose pore diameters range from 50 to 100 nanometers, smaller than COVID-19 virus particle size, which range from 120 to 160 nanometers. The Nanocellulose filter is made from plants, and one of the plants that has potential to be its raw material is water hyacinth. Water hyacinth has high cellulose content and abundant population. Therefore, this study aims to analyze the potential of water hyacinth as the raw material of nanocellulose filter cartridge. The output of this study is a review article that can be useful for the development of nanocellulose filter from water hyacinth. This paper uses systematic review with PRISMA guideline. We searched throughout Sinta, Indonesia OneSearch, Science Direct, and ResearchGate with the keywords water hyacinth, Eichhornia crassipes, nanofilter, nanocellulose fiber, water hyacinth population, and filter cartridge. We examined the eligibility independently. And we also reviewed cross-references cited in retrieved articles to identify additional relevant studies. We narrowed down the list of studies by selecting studies that examine water hyacinth nanocellulose filter, other plants' potential in making nanocellulose filter, water hyacinth potential for nanocellulose filter, studies published in English and Bahasa Indonesia, all experimental and observational studies until the date of search, October 2021, and we excluded case reports, letters, editorials, and opinions. Data regarding selected variables were extracted from each included study as per the objective of this review. The PRISMA flow diagram can be seen. We identified 433 articles and narrowed it down to 16 articles. The narrative summary approach is used for data synthesis, and research data taken from the results of the following review are presented in tabulated form. The data were categorized and tabulated based on review objectives. This is the result of the study. First, we'll look into previous researches on nanocellulose fiber filter. So the nanocellulose suspension is shaped into a sheet and can hold a small particles as the size of bacteria and viruses from passing through the pores of the mask. The filter also pay attention to breathability, which is a way to keep someone's breath comfortable when using a cloth mask. In this table, we can see experimental studies for similar nanocellulose filters. First one is conducted by Xiong in 2021, which stated that exhibited that nanocellulose made from a softwood bleach kraft pulp exhibited a high filtration efficiency of 94.6% for particle micron 0.3 and low pressure drops. And also Alexandrescu in 2016 stated that eucalyptus pulp nanocellulose filter exhibit filtration efficiency from 46 and 54% from two samples, and pressure dropped 55 Pascal and 25 Pascal. Review studies on the potential materials of nanocellulose filter for cloth mask on this table in 2022 by Rohmawati stated that coconut wire is potential for having around 54% cellulose content. And also Rimasari in 2022 stated that corn cob has around 66% cellulose content, and both usage of these materials have the advantages to reduce waste, and both materials have cellulose content higher than 50%. However, water hyacinth also has higher cellulose content than the two, which will be discussed next. So in comparison of the cellulose content and physical characteristic, there are water hyacinth, corn cob, coconut wire, Semarang kapok, Chinese kapok, cotton, giant milkweed, and also sisal. So by cellulose content, water hyacinth is the third highest after cotton and Semarang kapok, and the length of the fiber is longer than most plants mentioned here, but it's not as long as sisal, which means that it supports breathability but can still trap small particles. The diameter of the single fiber is also pretty small, and it has low elasticity, which can hinder the filtration ability of the mask. So along with the high cellulose content and great physical characteristic, water hyacinth has other advantages such as rapid growth. It duplicates every 11 to 15 days and normally cause biodiversity problems. And because of this, it has the possibility to be sustainable raw materials for nanocellulose because the population most likely will not decline. Also, because it is an invasive species, the use of water hyacinth helps to control the population without harming the environment, because to be able to be used as a raw materials, it has to be taken in a mechanical way, not in a chemical or biological way. So bottom line, water hyacinth has a great potential to be nanocellulose raw materials, and the use of it can also diminish the ecological and biodiversity problems that it causes. So we also want to talk a bit about the main steps in making nanocellulose filter from water hyacinth. First one is popping. The purpose of it is to extract the cellulose, and then we do bleaching to remove residual lignin, and then acid hydrolysis to increase the crystallinity of the cellulose. And after that, there is sonication to dissolve the nanocellulose fibers, and homogenization to reduce the size of nanocellulose fibers. And finally, electrospinning method is used to spin nanocellulose fibers into paper-like sheets. So to conclude the research, nanocellulose filter as an additional layer on a cloth mask aims to increase its effectiveness in filtering the COVID-19 virus during the pandemic. And based on the literature studies and comparisons conducted above, water hyacinth has a cellulose content above 50% and shows physical characteristics that have high potential to be used as raw material for nanocellulose filters. Albeit the lack of empirical evidence, innovation in the use of water hyacinth can have positive impact on the economy and ecology. However, further research is still needed. So here are the references we used, and thank you.
Thank you for the presentation, Nadia. It is a study literature, yeah, to have the future research. Maybe, is there any question to the other presenter, participant? Okay, it's not yet a question. A little bit question for me. That we know that Ecornia aggressivant is already used for furniture to make other furniture, table, something like that. So from your literature, is Acronia gracipan that will be used from raw material or for fermented or something like that, or you can use it in finding in the something live Acornia grassipens. I mean that like a rice house is the waste product, but Acornia grassipen is still live and invasive species. So what's your next research according to the literature you read?
I'm sorry, I have a trouble in my connection. Can you repeat the last sentence, Bu Reni?
Okay, what's your next research according to your study literature? Because Acornia gracipen is already used for furniture. Is there a lot of raw material to make filter culturite or mass, something like that? And the second, that we know that there is a lot of fiber. If we use this for masker, is there any pore for the fiber so that we can breathe healthy, or we can safe breathe with that raw material made by Acornia grassipens?
Okay, thank you for the question, Bu Reni. So the further research will be, I think, the what concentration of the water hyacinth is the best at making the filter, and it can be tested by the BFE, bacterial filtration efficiency, or other microorganism filtration efficiency, and also the pressure drop. Pressure drop is to test the breathability, and also for the raw materials, it used a fresh water hyacinth, and based on the literature review we conducted, water hyacinth is still causing some biodiversity problems, and I think it will be okay to be used, although it also used as a furniture. And also I know that we know that water hyacinth is very fibrous, but based on the steps on the making of the filter, the lignin on the water hyacinth will be diminished, so it's not really as fibrous as it used to be.
Thank you for the answer, Miladia. The time is up. Maybe we should applause to the Miladia's presentation. We will invite for the next presenter, Mrs. Aprilia, about effectiveness of cloud mass with active carbon filter from sugarcane bagasse. Siska, or one of the team.
Hello everyone, let me introduce myself. My name is Siska Aprilia Kaulika from Biology Department Universitas Negeri Jakarta. In this time, I will give a presentation of my research, and the title is Effectiveness of Cloud Mass with Sugarcane Bagasse Activated Carbon Filter. The current worldwide COVID-19 pandemic is caused by SARS-CoV-2 virus that attack the respiratory system. This virus has potential for rapid spread because it can spread through droplets. The high rate of spread of SARS-CoV-2 at this time caused the Indonesian government to require the use of masks for the entire community to reduce the spread of the virus. Use of mass recommended by government is proportional to amount of mass waste generated. Therefore, an appropriate solution is needs such as using masks that are environmentally friendly and efficient in preventing exposure to SARS-CoV-2 by using cloth masks and additional filters. In cloth masks play a role increasing the filtration ability of masks, especially in reducing microorganisms. One of the most common filters for masks is activated carbon filters. An activated carbon filter as a mask filter is due to increased ability absorption of microorganisms with amorphous property consisting mostly of a free carbon. One of most material that can be potential is bagasse. Activated carbon from bagasse has been widely used for metal absorbent and dye waste. Bagasse has the potential to be used as activated carbon because it contains lignin cellulose biomass with high carbon content. And the purpose of this research is analysis microstructure of activated carbon filter to analyze effectiveness in reducing microorganisms and to analyze user comfort level with differential pressure test. And the method of this research is use the true experiment, and the research design is used completely randomized design with one treatment of potassium permanganate with concentration of 5%, 10%, and 19 and 15%. And the data is analyzed with one-way ANOVA. And this is the procedure of this research. Is first with sample preparation, and next is 2-3 tasks. With first is scanning electron microscope with type of the microscope is SNE 15 5 5000 and 5400 m, and with um 500 apa ya 1500 and 10 and 1000 magnification, and the bacterial filtration efficiency with ASTFM F21015 standard, and with efficiency more than 95%, and the pressure drop using EN14683 standard with the differential pressure of less than 4.08 mmH2O centimeter. And results and discussion with scanning electron microscope activated carbon powder is in on these three pictures is we know that the activated carbon powder is generally dominant in form of irregular reticular fractures with more characters, and the density carbon has three different levels according to the concentration variation. Activated carbon with the highest density level is at concentration 5%. And the next, we analyze porosity level and surface area of each test sample is calculated with using following formula. Okay, and based on the results in table 1, it can be seen that the smallest percentage of porosity level is activated carbon powder with concentration 5%. And next, results and discussion scanning electron microscope of activated carbon filters. Okay, and the activated carbon filters is analyzed with diameter and length of the fiber of each test sample and is calculated using following formula. And based on the results on table 2, it can be seen that the shortest and the longest size is activated carbon filter is with concentration 5%. And the next is results of bacterial filtration efficiency. On table 3, we know that ANOVA test results on BFA, there is no significant difference in the BF result for each concentration. And the table 4 shows that the activator 5 with 15% concentration has the highest efficiency, which is 36.5%. And the next is results of pressure drop. In table 5, we know that the ANOVA test results of pressure drop, there is not significant difference between the 3 mass sample of each concentration. And the base of table 6, it can be seen that the smallest value in pressure drop test is 3.675 mmH2O centimeter, which is in the sample 5% concentration. And the conclusion of this research is activated carbon powder has a rectangular shape in irregular fractures, different density and porosity for each concentration, and the activated carbon filter size has a larger diameter than bacteria and virus. And 2, clot masks were considered less effective in reducing microorganisms with a percentage of filtration ability below 95% of ASTF MF2 101-14 standard. And the 3 only concentration of 5% has a value that meets the EN 14683 standard, which is less than 4.08 mmH2 centimeter. And this is a reference that I use. Okay, thank you for your attention.
Okay, thank you for the presentation, Ms. Siska. For the participant, is there any question? Okay, Siska, I will ask a little. According to the test, there is no difference between 2 samples for BFA. Yeah, this the results compared to another sample because for the control or negative treatment. So we know that that there is a difference between used in for your mass. I mean that you compare 2 samples for BFA and the data show that there is no difference between 2 samples. So is there a control one to compare a BFA so that we know that yes, I forgot the name of the ampas tebu in English. Sugarcane baggage, yeah.
Okay. Thank you, Bu Reni, for the question, and let me answer this question. And this BFA test is not use the control, but use a standard of blood mass as standard SNE, something like that.
Okay, is the data is below or above the standard?
Yes.
Below or above?
Above. I'm sorry. Below standard.
Below standard, so the mass could cannot be used for mass because this is under the standard.
Maybe can be used but the cloth mass is sorry, but the cloth mass material is must to be replaced at the other materials, ma'am.
Sorry, the next project maybe the team should edit some material to make
Yes,
it above the standard. Okay.
Yes.
Yeah, thank you for the answer. But according to the SAM, there is no difference.
Yes, ma'am.
Okay. Okay, thank you for the nice presentation for Mrs. Apriliani. We have to approach to the presentation. We should present Ms. Ariani Noviantari about review isolation of mesenchymal stem cell from human breast milk.
Bu Redi, I'm sorry, but Miss Ariani didn't submit the video and didn't attend the conference, so I think we can skip to Pak Agung Sedayu.
Okay. Okay, for the next presentation, the team of Pak Agung Sedayu, ABS 180. Oh,
Assalamualaikum warahmatullahi wabarakatuh.
I'm sorry,
Allow me to present our research entitled Two-Edged Sword, the role of alien invasive Muntingia calabura L for urban birds in Jakarta. This research was done by Agung Sedayu and Muhammad Isnin Nur. As we understand, that Muntingia calabura is an invasive alien species of plant across tropical urban environment throughout the world, including Jakarta. This species produce highly favored fruit by diurnal birds, and also in urban birds as in Jakarta. Urban birds species come to fruiting trees. Whether is it for food or for shelter? This is the thing that we want to explore in our research. If birds visit fruiting Muntingia calabura, is it only for food or shelter? And if the bird visit the Muntingia calabura, will the urban attributes, including area greenness or the tree posture, contribute to the visit? This is the thing that we will want to explore in our research. So we did an 68 research, 68 fruiting Muntingia calabura trees across Jabodetabek, and we observed the tree, each tree twice. We measured the tree dimension, the canopy sizes, number of ripe fruit, the ratio between the unripe and ripe fruit, and we also did location survey using Landsat 8. We measured the mean and DVI in radius 100 meters, and the tree we put it in the center of the radius, and we observe the bird directly in the tree 2 times in a week. We record species number, individual number, the number of individual feeding and non-feeding in the tree. The result was then analyzed by Spearman correlation, and furthermore, we redo the analysis using GLMM, or a generalized linear mix model, to evaluate the best predictor among many variables that we recorded. The research said that using Spearman correlation amongst the variable, we see that the number of canopy size and the location, the NDVI, are always not significant among all the among all other variables recorded, and the variables concerning the number of fruit and the number of the ripe or unripe fruits is always significant here. It means that the birds seems to come to the tree which produce more fruit compared to the tree that lives in a more green area according to NDVI, or the tree that provides more shelter or protection to the birds according to the size of the canopy. However, if we see the further analysis using the GLMM, and we make a different modeling, we see that the most probable explanation for the bird visit is not only the fruit, but also the canopy area, canopy diameter, and mean DVI. Canopy area and canopy diameter is the attribute of tree posture, and the mean and DVI is the attribute of area greenness. So the interaction between variables in the model reveals that location, green area or NDVI, and tree posture are good predictor for bird species, which can be seen in the highest AICC value. So, indirect relationship as the Spearman correlation. Urban bird seems to visit trees with fruits despite location and tree posture. However, in the context of urban environment, the GLMM models reveals that urban birds needs the protection from their environment even higher than the food source provided by Muntingia calabura. It means that as urban bird visits are not driven mainly by food availability, but more pronounced by environmental factor, green area and tree posture, which are also provided by other trees other than Muntingia calabura, it is recommended that the urban greening program discourage or further exclude Muntingia calabura amongst its tree list. I think that's our presentation. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
I think Pak Agung is already here and the team. Is there any question for the participant? Or Ibu Siti Nur Amaliati Priyono, is there any question? This is about the interaction between Muntingia and how many times the birds nest or invited the plant, maybe, something like that. Ibu Siti? Okay, maybe I will ask to Mr. Agung. What's the, according to the title, it's said that two-edged swords. What is the positive and negative of the Muntingia calabura for the urban bird?
Thank you very much for the questions. I put the title two-edged sword. It means that Muntingia can act as a producer, and it can act as a fruit producer for the frugivorous birds, and also as protection with its canopy. However, they are introduced from the South Americas. It means that they are a possible threat to Indonesian biodiversity. I put the word two-edged sword means that it is indeed providing birds the protection and the fruit as food. However, the bird that eats the fruit also contributes to the wide range of distribution of this species. It means that in the context of urban management, we should discourage the planting of this species in order to reduce the interaction between the plants and the birds to further decrease the spread of the species. I think that what we mean by the two-edged sword. Yes.
So they have a negative effect that if urban birds eat the fruit of Muntingia collabora, so they can spread the invasive model in other place.
Yes. That's the point.
Okay. Thank you for the answer, Pak Agung. If there is there's any question for the participant, we can close the Pak Agung presentation. Thank you, Pak Agung. For the next, and will be your presentation about variation of calcium oxalate rapid in several urban ornamental areas in Jakarta. Please, Pak Agung, to present this.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Allow me to describe our research entitled The Variation of Calcium Oxalaterophytes in Several Urban Ornamental Areas in Jakarta. This research was done by Agus Dayu, Mika Miarsa, and Matis Nur. As we understand, in urban environment as Jakarta metropolitan, the species that live in Jakarta, that is the biodiversity, urban biodiversity living in Jakarta, is highly managed by humans, including the plant diversity. So it is like that human decide which species lives in Jakarta or which species cannot live in Jakarta. Therefore, we understand by this capability of human, we have high diversity of useful plants living in urban environment, and especially the highest diversity of plant living in Jakarta is from ornamental plants, which serve as the adornment of our environment. One of which very popular group of the ornamental plants is the aroid family, or Araceae, which unfortunately comes from South America, many of them. Only several species are native to Indonesia. Among the urban arid, most of them are popular ornamental. Some are a source of wood, some are a source of medicine, but the most popular usage of the arid are as ornamental. However, all arids intrinsically having raphids as self-defense mechanism. So the raphids will pierce the human or any herbivore that eat or crush the leaves or stems or petiole and cause a burning sensation to the skin or to the mouth or any epithelious gland. And this rabbits is probable threat to human, especially to hobbyists, to enthusiasts, to gather people which is enjoying airwit as their hobby. During the pandemic, COVID-19, these past 2 years, the airwits are very popular. So in this research, we try to mitigate the danger of airwit because some people are very prone of having health hazard because of this rapid inside the leaves or the stem or even the roots of the airwit. So we need to understand the types, the location of the rapids across a range of species common living in Jakarta. And in these results, we expect to understand and furthermore to mitigate the threats to human, especially the enthusiasts and the hobbyists, which love airwit as their hobby. So we did something in 6 locations in Jakarta, and we found in garden vendor in Jakarta 8 most common species: Anthurium crystallinum, Anthurium promanii, Dieffenbachia amuna, Epiprenum aureum, Monstera adansonii, Philodendron burlamaxii, Synapsus pictus, and Syngonium podophyllum. And we survey among each of the 8 species the leaf, the petiole, and the stem, and we record the rapid deposited in those organs. We did anatomic resection in the lab, and the results are described in descriptive analysis and also discriminant analysis. We found that there are 2 types of rabbits within the 8 species, which are the stout shorter type, or we call the T1, a smaller one, averaging 57.72 to 72.37 micrometers in all 8 species. In only 3 species, we also found other than the stout shorter type or the T1, the T2, the thin and longer type rabbits. Average size can be twice as much compared to T1, 113.77 to 179.37 micrometers, but only in this species. When we map the T1 in the discriminant analysis, we found that the largest variation occur in Monstera adansonii, or very popular in the name of Janda Bolong in Jakarta, and the least variation of the rabbits occur in Dieffenbachia amuna, the green one. You can see this green one has a smaller and the least of variation, which we think it looks like the Monstera adansonii may perhaps pose a greater or higher threats to gardener compared to Dieffenbachia amuna. However, if we see the other type of rabbits, the 3 species, Syngonium podophyllum, Philodendron burlamaxii, and Dieffenbachia amuna, they have another type of rabbits, the T2. And because if we see the size and the formation of the T2, longer but thinner, and also with higher density compared to the T1, we believe that these three species might have a more hazard possibility since the T2 has significantly longer dimension compared to the T1. So we believe all 8 species are equipped with rapids, which means we must be careful in handling all 8 species. But among 8 species, we must be very extra careful into 3 species which have a probable higher hazard because they have T2 rapid types, and especially the Dieffenbachia ammuna, which have the longest T2 rapid types. I think that's my description. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for the nice presentation. For the participant, is there any question or discuss? Let's speak about the results. As we know that calcium oxalate is the waste product of the plant that can form in the vacuola of the plant. So is there any differences in the ornamental aroid ini if there are a lot of calcium oxalate found in the vacuola of the plant? I mean, can we describe the plant phenotypically that are a lot of calcium oxalate found in the different plant Araceae?
I try to understand your question. From what I see in the microscope section, that these rapids is everywhere. I cannot decide where they come from. If you say it's from the vacuole, well, it is very difficult to observe. Well, it should be there from the vacuole, and as it is written in the textbook. But seeing it myself in under the microscope, I see whenever I make a cut, the rapids will burst everywhere. Everywhere they will burst.
Yes, it is.
Yeah, I cannot decide where the origin of the rapids. So if it is from the vacuole, then I have to see further to determine that it is from the vacuole. However, from the observation that I see that the Araceae has a more common the smaller rapid ones, the small and stout one.
The length of
I haven't seen the terminology in the literature, so I just name it the T1 type of rapids, and the other one is the T2, which we found in only a few. So it means perhaps we should enlarge the survey and incorporate many more species, so we understand in Jakarta which species can pose more threat to people compared to this other species, which is proved to have a larger, longer type of rapids. I think that's a challenge for us.
Yes, it is that the calcium oxalate is the waste product of the plant, so they spend out their storage in the vacuola. But this is true that if we are cut the leaf, they spread in everywhere. So that can we justify that if rapid is spread, they're called a needle point calcium oxalate?
I'm sorry, I didn't catch your
rapid calcium oxalates is like combine or make it some of needle calcium oxalate we can found in many plant.
Yes.
Is there any confusion to describe where is it is the rapid or needle point calcium oxalate, or the team just measure the length of the rapid, something like that?
Yes. Perhaps to be honest, I think I should check other literature to understand more about types of rapids and types of calcium oxalate, because the 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 types that I found in other species, like I just tried a species comes from Sulawesi, it is very different from needles that I saw in RACE. So perhaps I should make it better in classification to reclassify our research. I think we are open to suggestion.
It's very interesting. One more question, that is that the conclusion said that the 3 species from Arase found that this is hazard for the human. Is it true?
So far, yes. So far, from 8 species, we have 3 species which has different kind of rapids. I think seeing the size and the formation, we can justify that they perhaps has more threat to human. But of course, we need to do a human test, and we didn't do that so far. Well, I need more volunteer perhaps.
Okay, that Arase is not for consumption in human.
Some of them, yes. Some of them, taro, very common. Taro, very common.
The leaf of the taro.
Yes, the
Okay.
rhizome and the leaf.
Yeah.
And petiole is very common to be used as vegetable in Indonesian culture, Indian culture, or Pacific Islanders.
Yes, that's very interesting to found another rapid calcium oxalate in vegetable or something like that.
Yes, I
This
think
is very interesting,
I should add that people traditionally has tradition way to cope with the problem. Indian and Indonesian, they use heat, they use pounding to break the rapids. But in our context, this research is done to help people, hobbyists, people with enthusiast to gardening, because they don't eat the RSE. They don't eat the RSE. So perhaps their hazard comes from touching, not from eating.
Yeah.
Yes, yeah, that's our concern with this research.
Okay, thank you very much, Agung, for the nice presentation. I will give the presentation session ini to the Miss Miladia Samsu as MC or PIC in this room. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Miladia will be informed to the participant.
Okay, so we have reached to the end of the session, and you may turn on your camera because we will take a second photo session. Okay, I think there aren't anyone who want to turn on the camera, so I will take the photo. Okay, 3, 2, 1. Okay, and before we close the session, I want to inform a bit. So the certificate will be able to be generated through registration system on September 9th, 2022, and information of the best presenter will be able in the website, on the conference website, on the same day as the certificate. And also, please, if you haven't uploaded your full paper to the system, please upload it so you can complete the publication process. And also, publication payment only paid when paper has been accepted. And I would like to thank all of you because you already participate in this conference. You may leave the meeting. Thank you.
Thank you very much. See you in the next seminar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.